Welcome back. Let's begin the next part of the program. I'm Åsa Pedersen. I work at the Norwegian Environment Agency at the section for climate knowledge. And I have the pleasure to guide you through the rest of the talks until the discussion. And the first talk will be given by Tina Kutti. She's a benthos ecologist with a PhD from the University of Bergen. She's worked at the Institute for Marine Science for 3 years, and she's done a lot of research on corals and ocean acidification.
Okay, thank you. I've been given the honor to come here today to present what IMR is doing related to biological effects of ocean acidification. This is what people have sent me. So it's not representing everything, but a big proportion of what has been done at IMR recently. So I'm not going to talk about old work, but new things. First, shortly about laboratory experiments, some results regarding copepods and fish larvae, lobster and coral reefs. There's also ongoing experiments with mesocosms. That's a collaboration project with China to look at food security, future aquaculture production. I'm not going to mention that later. Some new multiple stressor effects that we're starting. We have adapted sponges and corals to high CO2 and high temperature for several months now, and in a month we're going to expose them to different types of sediment to look at the effects. I'm not going to talk more about that either. I'm going to discuss the work that we've been doing at IMR looking into natural gradients of CO2 and how they can help us understand the biological effects of ocean acidification. Some work that Sam Rastrick has been doing at Dominica. Also some new wall reef studies that we're going to carry out this autumn. And also I'm going to present this new ESSIS working group. All right. Howard Brauman, together with a lot of colleagues, see here the list, has been working on the effects of high CO2 and temperature on copepods and fish larvae. They've been very thorough, looking at a huge range of biological endpoints to try to understand how ocean acidification will affect these organisms. They've been looking at development rates and growth, molting success of the overwintering copepods, lipid sac size, different composition of elements, metabolic rate and cellular metabolism, global proteinome and gene expression, and carapace integrity, fatty acid profiles. They've also been looking at different behavioral responses, like predation, swimming behavior, but also feeding. So a huge amount of work. The outcome of this work is that they found no negative effects on cod and herring larvae, no negative effects on Calanus, the different Calanus species they've been looking into, or salmon lice.
You're very disappointed in that, I must say.
And possible beneficial effects on Calanus finmarchicus on growth and lipid content, but only when temperature is not elevated. So that's good news for these species. Turning into the lobster work, and Lisbeth Agnalt and co-workers have been doing new experiments this year. Not so good news. High CO2 and high temperature increases the occurrence of deformities in lobster larvae and juveniles under optimal feeding. They see no effects on the high CO2 on the size of the larvae and juveniles under optimal feeding. But when feeding is reduced, all these high CO2 scenarios, no larvae survived until metamorphosis. So not so good news for this species, but a little bit dependent on food availability, which we know very little about in the future world. Some new results from a 1-year exposure experiment that we've been doing on cold water coral reef organisms. We're just about to sample last time. We just started, had not finished yet. But what we have observed is a high mortality of bivalves and sponges that live in the coral reef when they're exposed to high CO2 and increased temperature. And they started dying already after 6 months. But now in our high CO2, increased temperature treatments, we have no bivalves left. The corals do much better. They survive in these treatments, but show a reduced growth. Interestingly, the corals that live in a starved environment, exposed to high CO2 and temperature, died. So even though we know that the cold water corals can absorb dissolved organic matter, this is not enough for them when they're exposed to this high CO2 and high temperature. We also seen that bioerosion is enhanced under high CO2, which means that the dead framework that supports the reef will be removed quicker. And this is particularly high under increased feeding conditions. So for the cold water coral reefs in Norway, the situation doesn't look very good. Okay, so moving on to a different approach to look at the effects of ocean acidification using natural analogues. Sam Rastrick and co-workers have been studying some CO2 vents in a Caribbean island, Dominica, where CO2 is released into the water, and studied the community structure along this gradient. You can see the different pH levels along the gradient down there to see who will be the winner and who will be the loser in the future world. We've also performed some reciprocal transplantation experiments, both short-term and long-term, on corals and sponges to see the physiological response of the organism to the changing environment, and also how to assess their capacity to adapt to this new environment. Why do we want to look at natural gradients? It's because we can study the capacity for adaptations and acclimatization over generations, which is difficult to do in laboratory experiments unless you have very short-lived species. We can also look at community-level responses. We can look at how the community structure will change and also how diversity will be affected. And we can scale what we see in physiology to the ecological level. So we will incorporate things like also changes in food quality, competition between species, and also other interactions. We recently got some extra money from Miljødirektoratet to analyze data, abiotic data that we have been collecting from wall reefs in Hardangerfjorden. We now have data from spring, late summer, and winter for 2 years. And we want to use that to see if we can correlate it to the vertical and horizontal distribution of corals in the fjords that we've also mapped. So that's going to be very exciting. We've also, this summer, formed a new working group in ESSAS called Analogues of an Arctic in Rapid Transition. And the aim of this working group is to identify suitable areas where we can use these natural analogues to try to predict how ocean acidification will affect the ecosystems in the Arctic and subarctic. So we're trying to identify suitable areas, which could be CO2 vent sites, front systems, areas with freshwater runoff, areas of sea, ice formation and melt, things that could affect the carbon chemistry. And we want to form a strong group that can work with this. And if anyone is interested in joining this group, you should contact Samuel Rastrick. Samuel, please give a wave. We're very interested in more people joining that group. If anyone can read very quickly, this is what the group at IMR has suggested that we should focus on in the future. Continue the good monitoring, but intensify the effort on natural variability, monitor biological effects, try to look at natural gradients, continue with the lab experiments on key species and also species with short generation times, multiple stressor experiments, trying to understand the mechanisms that drive the responses, look at indirect effects through trophic interactions, and also examine ecosystem effects through ecosystem modeling. That's our aims for the future. Thank you.
Thank you, Tina. We also want to give you a small present.
Thank you.
And next we have two speakers from the Norwegian Polar Institute. It's Agneta Fransson and Thor. And they are free to divide the time between them as they choose. Agneta is chemist with a PhD from Gothenburg University. She's worked at the Polar Institute for about 6 years, and she's one of the founders of the Fransson flagship on ocean acidification, together with Thor. And Thor is a marine biologist with a PhD from Copenhagen, and he's worked a lot on zooplankton and ocean acidification.
Yeah, thank you for the introduction. This is a work in the Fransson Ocean Acidification Flagship. And it's me and also Melissa Chierici, she couldn't come today, unfortunately. But we also have a new member that I've forgotten to write down, is Elizabeth Jones from IMR as well. She's here today, so she also contributed to this work. So we work in the Arctic and in Svalbard fjords, and we also look into the implication for calcifiers. How do I change slide? Or how do... Oh, okay, yeah. Okay, so the flagship ecosystem effects and trends of ocean acidification in Arctic waters was established in 2012. So now we have been working for this a while, and this flagship is divided into 4 compartments. It's the field study with observations to get time series and to find trend analysis, and also look into processes of the carbonate chemistry and ocean acidification. We have one part which is more biological effect studies in laboratory mainly on different organisms, and Peter will talk about that. Then we have a modeling part which focus on ecosystem modeling and also socioeconomic modeling to improve projections in future ocean acidification. And I'm more into the carbonate chemistry and observations, and now we have been collecting lots of data from 6 years now in different areas in the Arctic, and we collect for the whole carbonate chemistry, but we also collect other parameters such as nutrients, oxygen 18, that we can get fresh water and information, and we also of course have the physical salinity, temperature and everything. So we try to map everything in the water column in different areas. Marked in blue here, we have different transects. We have a Fram Strait annual expedition between Svalbard and Greenland, where we in September, August-September, and we have also the annual MOSH cruises, where we also have biological sampling as well, and carbonate, of course. And then we have Twain, and also Svalbard, and we also have the NICE. In 2015, we had this seasonal study, drift study from January to June, where we got data, interdisciplinary data from lots of parameters north of Svalbard and into the deep basin of the Nansen Basin here. And we also have a Barents Sea study as well. And then we have the red dots, which are more the Norwegian Sea Monitoring Project, which you already heard about. Now we have a smaller time series, and we work on it in Framstrait, and this is just an example how it looks like 1 year when you're in August, September. And this is Svalbard on this side and Greenland on this side, and we can see there's a large variability in the saturation, aragonite saturation. On the western side, we have the outflow of cold Arctic water, and you also have the lower aragonite saturation. And this cold water meets the warmer Atlantic water here on the eastern side, where you also have higher aragonite saturation, and that's an inflow to the Arctic Ocean. So it's a really large variability horizontal, but also you can see also vertically in the deep, down from surface to 400 meters here. And we study all physics, chemistry, and biology. We have also studies in Svalbard fjords. We go there every year, so we have 5 years of data now. This is an example of Kongsfjorden on Svalbard, and we studied both in winter and summer to look into the seasonal variability, but we also have different years. And Kongsfjorden has a glacier in the inner part of the fjord, so you can have meltwater there at certain times of the year, and you also have Atlantic water from the other. Here's a map. You see the Atlantic water, and you have this is a fjord, and you have the glacier in the inside. So that affects the chemistry, of course, and then the biology. So what we found, there was a very different situation between 2 years. And we have also more years, but this just shows the really contrasting years when we have, in 2013, we have near the glacier, we have lower saturation compared to summer 2014, where we have higher. And that we explained with it was more fresh water from the glacier in 2013 than in 2014. And it was more Atlantic water influence in 2014, so it depends a lot on the different water masses. And we have also lower saturation at the bottom of the fjord. So we also in the fjord study and try to collect the Limacina helicina, which is a theropod, and I will show more about that work. What we found in this fjord, in Kongsfjorden, was that the winter situation had the lowest saturation of 1.5, and you know that between 1 is a really bad situation for the calcifiers, and 1.5 is not so bad. But if you look into historical data and studies by Benashek and others, they found that the critical situation for this to form shells, if you reach 1.5 of saturation. So if you look at the gray area here, is the critical for the theropod or Limacina helicina to create shell, or they found deformation or thinning of the shell if you go into this critical zone. And for the Kongsfjorden, we are up here now, but with increased freshwater fraction here on the x-axis, if you increase freshwater by 1 or 2%, we will reach or approach this critical area where you actually have problems with the shell formation. So that was one of the results from this study we published last year, and we look into this theropod, Limacina helicina, because it has a large role in the Arctic ecosystem as food, serves as food for polar cod and seabirds, and also some whales, and it contributes with 20 to 42% to the global carbon carbonate budget. And it has this shell of thin shell of aragonite, so it's very sensitive to low pH, low aragonite saturation, and high CO2. And that in particular in combination to high levels, or high increased temperature, which has been found in studies by Comeau and Lischka, who did a study on theropods in Kongsfjorden. And Banaszek and others, they found that, as I said, with below 1.4, you found thinning of shells, and even lower, you could also find even worse, they can't produce it at all. But that study was performed in the Californian coast and also in Antarctica. Theropods also have a journal, vertical migration, and is important for the exposure time and duration, when it moves down and how long time it stays. And juveniles decide in winter, low food concentration. Yeah, and Limacina may be a suitable indicator for ocean acidification effects. And this Mano et al. Summarize, it's a paper came out this year on shelled theropods in peril, assessing vulnerability in a high CO2 ocean, based on the workshop in 2015 when I and Melissa took part, and also in this paper, summarizing research on theropods. And there was a workshop in 2015 here at Miljødirektoratet, and also a report came out where we discussed the possibility for Limacina helicina to act as a indicator, biological indicator. So we work on, in this flagship, we work on sampling, continuation, development of techniques, and evaluation. And here are some studies we did last summer with sampling of theropods. We used nets, and we found lots of juveniles in fall, and that suggests that they are recently hatched, or we couldn't find them, the big ones. So there are some questions still. This is around Svalbard. And we also find a warm or less cold-adapted species, Retroversa, which also have this shell, but this is a smaller one. And that could also compete with the Limacina helicina, which is a cold-adapted. So depending on the water masses, you could find different species here. I will just show you the method we use. This is a Japanese lab in Jamstek, and they have this fancy instrument where you can measure the shell density and thickness using micro X-ray tomography, and we relate it to carbonate chemistry in the environment. So this you can see this is a shell. And the color here is the thickness or porosity of the shell. So this is something we still working together with the scientists there. And that's just for the discussions. So maybe I don't have to say this longer, but this is for the discussion. Can we use the theropod as an indicator for ocean acidification? We have been questioned that, and they are sensitive to low aragonite saturation. That's true, but can we monitor them? This is a challenge because of the patchy distribution, and we still lack knowledge about the life cycle and how they behave and respond to ocean acidification and other stressors. And also the different life stages respond differently to ocean acidification. So we need more development of techniques, and also more research both in natural environment and in laboratory experiments. So that was it. Yep. So I think we will discuss this more in the afternoon.
Yes. Thank you, Agneta. And now Peter Thor will give a presentation on more biological effect research.
Okay, thank you.
Yes, the biological effects of ocean acidification in the Arctic. We've been working quite a lot on that in the Polarinstitutt, and I am sent to tell you a little bit about that. This is the same flagship that Agneta was showing you a little bit about. These are the different work packages, and the biological work or the effects studies were done in, which one is the pointer here? There it is. This is the work package 2 that's led by Howard from IMR and our very own Håkon Hope. This is basically what we have chosen to study. This is a very important species in the Arctic. It's one of the keystone species in the Arctic. It's very abundant on the shelves around the Arctic here, and it's a major grazer of phytoplankton. It contains a lot of lipids, many of these omega-3 lipids that we all enjoy to drink and eat. They're the primary prey for larval and juvenile fish. Not only are Arctic copepods the primary prey, but it's also on a global scale. Copepods have that sort of major ecological importance. But they're also important prey for whales and also some seabirds. So there's been a lot of studies have been conducted on this species. Also, people from RV in Germany has conducted a lot of physiological studies on Calanus glacialis. These are some of these results. They've been measuring ingestion rates and this is Calanus phlegmaticus and other species, and they've seen that there's pretty much no effects on ingestion rates here. Also, metabolic rates on these copepods have shown to be unaffected, but nevertheless, we've pressed on and tried to sort of dig deeper into the physiology of these copepods to see what if they are simply if they simply couldn't care less about ocean acidification, or if there's some kind of a physiological buffering going on. This is a study conducted at Østervold in Howard's lab on the development of the Norpiae larvae, also showing sort of this similar developmental rate regardless of the PCO2. So again, it seems like that this species is relatively or is tolerant to ocean acidification. But what we have also done, we have, as I told you, we've sort of digged a little deeper into the physiology, and this is a really weird picture. I'm not going to explain all of it to you, but this just simply shows the up and down regulation of the gene activity in these animals, and it seems like there is changes based on pH that are related to something called the universal cellular stress response. So it seems that they can maintain growth on the same level as without ocean acidification, but they do that in an active fashion. And you can then hypothesize, well, will they be able to do that for a longer period of time? We saw that in this experiment, they are actually able to do that because they can go through all these different stages that they go through their development without showing any effects. So what we also have done is to try and that hasn't been done before, roam around a little bit in the Arctic to see if these changes would vary from region to region, from population to population. Would there be the possibility that different populations would be adapted to the local environment that they live in? So we went to 2 different fjords in Svalbard, Kongsfjorden and Billefjorden. And Agneta just showed you the pH profiles of Kongsfjorden. Caught the copepods, took them to the lab and did our magic. Same thing for sort of a reference in the Disco Bay in Greenland, and did exactly the same thing. So it's kind of a common garden experiment without a common garden in a way. And this is what we found. We were able to study the effects on 2 different stages, two different larval stages. And the first pictures I showed you, with no effects in grazing rate and metabolic rates, all of these are conducted on the very same copepodite 5 stage, simply because the copepodite 5 stage is the stage that has the longest duration, and that is the stage that is typically there when you go into the Arctic, right? So there's no effect on this. But this is what we then came up with for the stage just before that. Again, ingestion rates or grazing rates and metabolic rates. And in the Disco Bay, it seems like these copepods, they are fairly tolerant to changes in pH. And this is very low pH, 6.5, right? Extremely low. So we've taken this kind of ecotoxicological approach where we want to have sort of a positive control as well, trying to push the envelope as much as possible. In Kongsfjorden, we got a significant decrease in the grazing rate and a significant increase in metabolic rate, and the same thing in the copepods in Billefjorden. This is the exact same species, the exact same larval stage, and at the same year. So what is the conclusion from this is then, if you take and calculate something called the scope for growth, you take the ingestion rate and multiply that by the absorption efficiency in the gut. So that's basically how much energy you can put, the rate of maximum energy you can put into your body, and then subtract the energy that is lost by respiration. Then you get something called the scope for growth, and this is the development of the scope for growth in these 2 different fjords. So it becomes negative at some point, 7 here and about 7.5 there. And that's not very good for anybody to live under these conditions. It won't last long. Even though this would occur only in one single larval stage, or perhaps 2, we don't know about the preceding stages, it would really have significant and severe effects for the populations. This is where we would expect to find ourselves in the year 2100, and we would still get, in Billefjorden, we would get a 50% drop in this scope for growth. So those are severe effects. But this is sort of to sum it up, what we know now for this particular species, and this is the whole life cycle of this species, summing up all of these different studies that has been done with no effects on egg production, egg hatching, lower survival, but we didn't see any effects in development of the larvae, the Norpgii. There's some effects on the feeding, metabolism, and this severe effect I just showed you. But in the C5s, they are fairly tolerant. So the picture is that, if you look at the whole big picture, they seem to be somewhat tolerant, but there are specific points in their development that could be tolerant. Okay, so just to sum it up, I also want to show that we do actually what Ara was sort of hinting about earlier on is that all of these effects may be masked by adaptation in all these species. People have been studying that. We have also been studying that in another species, a smaller species. This is egg production in this species at increasing PCO2. So there's a drop in 67% if you just take these copepods and incubate them right away. What you can then do is you can take these copepods and incubate them for a longer period of time, several generations, and what happens then? You only get a 29% drop in egg production rates. Right? So there's a transfer of resilience or tolerance among the generations or from generation to generation, and we now know that transfer is also accompanied by selection in certain genes. Right? So it is adaptation. So that's pretty much what we have done. What we would like to do with the flagship is also sort of to connect everything together, these biological effects, take those and put them into some these models, some of those that Kai was showing earlier, and connect that to the chemistry, and then eventually sort of with the help of people from Norge, get some kind of a socioeconomic consequence of this, because these copepods are really very important prey for fish larvae. So that's pretty much. Yeah, I can also say that this is also pretty much what we are trying to do in the Arctic monitoring and assessment program as well. Thank you. 0 minutes.
Thank you, Peter. A small gift for you.
Oh, thank you.
Next, we will move on to consequences and effects on the society. And it's Grete Hovelsrud who will give the talk. She is a professor in environmental sociology at Norsk Instistutt. She's been working in the Arctic as a researcher since 1981. She's also been an author in the IPCC fifth assessment report, in the working group 2 on impacts and adaptation. Yeah. And she's a co-leader of the research project Acid Coast, and that's why she's here today to present.
Thank you. So we're switching gears totally here, and I think it's interesting that this is the only. Just a minute. This is the only project that talks about the social side of things, and it came from many meetings with Richard Bellaby in Lofoten. We were all invited to these town meetings to talk about what's happening to fisheries and coastal zones and ocean acidification and so on in the Lofoten Islands. And finally we got a project together. We call it Acid Coast. And we are talking about co-production OA knowledge for adaptive co-management, in particular the coastal zone. One could think of the high seas, but we're starting in the coastal zone. So we would like to understand how the coastal zone governance and management can effectively respond. And that's something we don't really know, because it's something that we don't really see. And we don't really know how it affects the coastal zone. But we are sort of slowly moving along here in this, and we are trying to work, well, first of all, to understand how OA may impact coastal areas in Norway, and how we could make it a governable issue. It's very hard to govern climate change. It's even more difficult to govern ocean acidification. In particular, when you think about development of boundary arrangements, and I won't have time to go into boundary arrangements here, but I'll give you a little teaser for that, for how you can co-produce OA knowledge. And that may sound a little odd to most of you who actually go out and measure and have a very particular way and many samples. But there is a way to bring in different kinds of knowledges. That's why I love sitting here and listening to all this. I'm learning something, not in depth, but I start to understand. And I really enjoy the presentations, for my own sort of learning curve is going up. And then we're talking, when it comes to climate change and other areas, we talk about adaptive co-management. And we are looking into how that could be used for governing ocean acidification impacts. So we are studying existing coastal zones management in this project as well. And then we have our own little social science bit here. We would like to contribute to new models for adaptive coastal management. Just very briefly, we all know that the coast is not simple. There's a lot to govern, there's a lot to manage. There are lots of interests and lots of conflict of interests here, just to illustrate, and this is probably not all of them. So we are, the ocean acidification and the governing of it has to sort of fit in all of this. There are economic sectors, there are leisure activities, there's all kinds of activities. And we have, just very briefly, we have many public coastal zone management actors. And in a way, on paper, there's an aim to integrate coastal zone management, but it seems to be stuck in silos, nevertheless. We are looking into this. This is just a list of the different public coastal zone management actors, and you can see almost all of them approve permits in some fashion or another. So we'll tell you next time how this plays out. But this is just an illustration of what we have to deal with when we are trying to think about managing the impact of ocean acidification on coastal zone activities. When we've found, I've been working with climate change issues for many, many years, and we see that it isn't enough to just bring in more knowledge. It doesn't lead to action. We need more knowledge from a scientific perspective, but we may need to think differently how we communicate that knowledge. We see that the response to, that was the first one. The new insights generated by science do not automatically result in action. We know that. We know, those of us who've been working with climate change issues, there is something happening in that gap. So what we have been working with is to talk to people that are using the knowledge. It could be a local fisher, local organization, there could be managers, there could be anyone, but it has to be credible for them, and it has to be salient, it has to mean something and legitimate. That's difficult as a scientist to do, because we have to stick to our disciplines. So we're thinking about this area right here, this boundary work area, where there is something in that space. There's a lot of things to be done between the marine science and my science and the management. So we are looking in this project into a little bit on the communication, translation, and mediation of ocean acidification knowledge. What you have presented today is great science, and I've listened long enough to pick up things, but you can't present that to users out there, to managers. They would fall asleep, very fast. I mean, honestly, it has to be mediated somehow. It has to show why is this important. And when when Tina was talking about how it affected the lobster and not the larvae eggs for cod, I woke up, because we are very interested in knowing how the biology will be affected, and also the Calanius and the other whatever pods you were talking about. It's those things. If we could just lift that up and say, look at this, and be a little bit daring on how we communicate that knowledge further. So since I've been talking to Richard Bellaby for years about this, and we were like this far apart, and we moved, and we now understand each other. And Kai here, he's been to our project meetings. He knows that we can talk to each other, right? Sort of. Certainly after a few beers in the evening. We have these cases. It's kind of different when you have a social science case, because you can't just go in and sample a little here and there. You have to really go deep into the community. I've been working in the Lofoten area for many years, and the team on Vestlandsforskning knows Sunnhordland. So we have those two cases. There are two fjords there that are relevant. There are people, stakeholders, if you will, users who are interested in this knowledge. And then we have some very good partners at Oban SAMS in Argyll, or it's actually Oban, but same difference. So some of the methods we use is stakeholder involvement. To me, it does sound a little fluffy to me to say that. It sounds like, okay, tick off that box with stakeholder involvement. But we're actually doing it. We're actually sitting down with people, and we have had multiple workshops and data sampling by the users, which is something that Richard is taking care of. And there are multiple interviews and lots of discussions about the areas that are important to people. And then we have the OA measurements and modeling in these fjords that are very, very valuable for the people who live there and for management issues. We've done an assessment of current coastal management touching upon the ocean acidification, a literature review. I will show you a few of the results, and you will see that this is a huge topic. And we have knowledge exchange with Scotland through workshops. So it's a very sort of, I'm an anthropologist, and that's what we do. We understand the whole picture. What has been identified, some of the issues and challenges identified through the 3 stakeholder workshops that we've had to date, is how to identify the case study sites. By what parameters? Whose voices? Who decides what's important and why? And that's an interesting discussion to have. And it's from, if you then move maybe to some of the managers, you talk about the importance of piggybacking on the regional water directive work. So we don't reinvent the wheel or do something that it's good to harmonize with other work that's going on that is relevant. So you see, I mean, this is the stuff that comes up when you talk to people. Prospects for kelp production should be investigated. And somebody talked about the kelp case here in Solrun. Solbergstranda. And the whole issue of establishing regional coastal zone plans. These are issues that are coming up in the context of ocean acidification. The aquaculture industry are very interested in knowledge about OA impact, in particular with regarding to the wild salmon smolt. I don't know if anyone here works on that. I'm looking at Tina back there. Anyone work on that could provide some information? There are projects here for all of you guys as well, that from what we are finding out. Sometimes it's nice to actually develop projects that are based on some question that someone else is asking, that are concerned about a particular issue. That's one way to drive the research. Water sampling cruises, August 2016, and then we in Vestvåg June. And these are some of the data that we got from Richard. We're going to have a meeting in Lofoten next week, and I will get more discussion with him about all of this data, what it means, and how it all worked out. You see the shallow. They were measuring at shallow depths, which is, somebody talked about how it moves up in the column and that you see more and more trends for higher up. I don't know if what this means in terms of the whole increase of ocean acidification. Okay, very quickly, because I know it's lunchtime. This is some of from the literature that came up about coastal management and ocean acidification. There are lots to do. We need to raise awareness and increase the knowledge. That's the general category of action. More research into the impacts, and we have learned when the data that you present here are published, then we have learned more about the impacts on significant species, for example. Educate the planners, outreach activities to a broader audience, try to explain, and there are some really neat ways to illustrate, I think, ocean acidification. And of course, the mitigation side, reduce ocean acidification, that's easy to say. Stabilize, oh yeah, we all want to do this, of course, atmospheric CO2 levels. Where's the pointer? There. And there's a whole sort of group of mitigation activities that has been identified in this literature. I won't go into those because I'm sure you all know those. But the adaptation side is interestingly pointing to reducing other environmental stressors. That would be interesting to hear from you if that actually has an impact on, if that makes a difference. I'm looking at Peter there because he's nodding at me. That's always nice when someone talks back. Enhanced wastewater treatment and protect particularly sensitive coastal areas. We don't know exactly how this will play out in the coastal areas that we are working in. I thought this was interesting. Include potential OA stress in harvest decisions in fisheries management. Is that taken account of when we see how it affects the actual food for the fish? It's a big black box, and I'm really glad we could get this project because we are sort of starting to peek into the box at least. The restoration activities, of course, this is partly also from the southern ocean. Reduce overfishing. This is about strengthening the ecosystem resilience to meet the ocean acidification. Maybe it won't result in very, very challenging effects. Who knows? Adaptation in industries. Adopt flexible planning is something I think is important in any case. But we will publish this literature review with an analysis and we could send it to you. But what we're doing now is to establish user water sampling routines and initiate these so that people that are living there can not just Hurtigruten nor these box ferries or what they're called, but also the local coastal users that live there. And then we have the stakeholder feedback workshop on the first OA scenarios next week. That will be exciting. We have one on the west in Sunnhordland, in Kvinnherad, and one out in Leknes. And then we are of course working on publications, and the Scottish Knowledge Exchange Workshop in November. And so we will come equipped with the feedback from our workshops with the stakeholders here when we go to Scotland in November. Thank you. I suppose I don't have time for questions. But I did leave. Who's Howard? Howard? I sent you some questions for your discussion points.
Oh, I didn't see them, sorry. I don't know when you sent them, but
I sent them on email this morning, but I'll give them to you.
We can talk about this in the lunch break.
Great.
I think we can give you 2-3 minutes for questions, if anyone has any questions. Or we save them to the discussion later on today. Thank you very much, Grete, for a very interesting talk that brings the ocean acidification problem into a more society perspective. This will be very important for the continued program after lunch. First, we will have some talks. They will be in Norwegian. So after lunch, we switch to Norwegian. And then for the discussion, we will start a bit in English, and then we will use both languages as we please. But now we will have a lunch break until 12:30. And the lunch will be served right outside the doors. Thank you.